Hi everyone, welcome back to my channel. As a continuation part of cell cycle and cell division, in this video I'm going to talk about the concept mitosis. Mitosis is a cell division process in which the daughter chromosomes are distributed to the daughter cells equally. You all know that DNA replication or the synthesis of DNA takes place in the S phase of the cell cycle, and this replicated DNA are distributed to the daughter cells in the M phase of the both mitosis as well as meiosis. Anyhow, in this video I'm going to just talk about the mitosis. In the mitosis, there is an equal distribution of the chromosome to the daughter cells, due to which the number of chromosomes in the parent cell as well as in the daughter cells remains same. That is why mitosis is also called as equational division, because number of chromosomes are distributed equally to the daughter cells, or the number of chromosomes in the parent cell as well as in the daughter cells remains same. This mitosis process takes place in the body cell or the somatic cell. That is why it is also called as somatic division. As I told in the last video, M phase takes place in two steps, that is karyokinesis and cytokinesis. Karyokinesis is a process where the division of nucleus takes place or separation of the daughter chromosome takes place, whereas cytokinesis is a process where the division of cytoplasm takes place or distribution of the cytoplasm to the daughter cells takes place. We'll discuss these two steps one by one. First one is karyokinesis. As I already told, in karyokinesis, division of a nucleus takes place. Karyokinesis is divided into mainly four stages for our convenience. Anyhow, karyokinesis is a continuous process and you can't draw a clear line between the different stages of the karyokinesis. But our convenience, we recognized four phases or stages under karyokinesis. They are prophase, metaphase, anaphase and telophase. You can see varieties of changes or remarked changes in the chromosome as well as in different structures of the cell in all these four stages. We'll discuss all those changes one by one. First, let me talk about the first stage, that is prophase. This prophase stage begins as soon as the interphase is over. You all know that the chromosome in the S and the G2 phase of the interphase is long, thin, thread-like, and this thread-like chromosome start to coil or folded to become a thick and short chromosome, and that process of coiling or the folding is known as condensation. So condensation process begins in the prophase stage. You all know that the chromosome is made up of two threads or two chromatids, and these two chromatids are held together at the point called as centromere. So these two thread-like chromatids start to undergo condensation to become short and thick mitotic chromosome. These are the changes that takes place in the chromosome in the prophase stage. Apart from that, some changes takes place even in the centrioles. You know that the duplication of the centriole or centriole replication takes place in the S phase, and this duplicated centriole start to move apart and it moves towards the two poles of the cell in the prophase stage. Some delicate fibers called as spindle fiber is also formed between these centrioles. Apart from that, the nuclear envelope which is present around the chromosome start to break down and it finally disappears. The nucleolus also start to disintegrate and it finally disappears. So these are the changes that takes place in the prophase stage of the karyokinesis. Now let me talk about the second stage, that is metaphase. In the metaphase stage, due to disappearance of the nuclear envelope, the chromosome spreads in the cytoplasm. In this stage, the chromosomes are highly condensed to form with the short and thick chromatids, due to which the chromosomes are clearly visible under microscope. As you all know, in the metaphase chromosome, it consisting of short and thick two chromatids which are held together at the point known as centromere. On the surface of the centromere, it has a small disc-shaped structure called as kinetochore. So, kinetochore is present on the surface of the centromere. This kinetochore serves as a site of attachment for the spindle fiber. So, some of the spindle fiber attaches to this kinetochore of the chromosome, and they bring the chromosome to the center of the cell or to the equatorial region. This movement of chromosome to the center of the cell is called as congregation. So, the metaphase stage is characterized by the movement of the chromosome to the equatorial region or to the center of the cell in such a way that the one chromatid is attached to the spindle fiber of the one pole, whereas another chromatid is attached to the spindle fiber of the another pole. As you can see in this diagram, the purple chromatid is attached to the spindle fiber which arises from the centriole of this pole, and the pink chromatid is attached to the spindle fiber which arises from the centriole of this pole. So the chromatids or the chromosomes align themselves in the center of or at the equatorial region, or they align at the single plane called as metaphase plate. So that is about the metaphase stage. Now let me talk about the third stage, that is anaphase. In the anaphase, the chromosome which was present on the metaphase plate or at the equatorial region or at the center of the cell now start to split or separate, and now those chromosomes are called as daughter chromosome. After the separation, one single-stranded daughter chromosome moved towards the one pole and another single-stranded daughter chromosome moved towards the another pole. As a result of this movement of chromosome away from this equatorial plate, the kinetochore of each chromosome will be towards the pole and the arms trail behind it. As you can see, these are the kinetochore which move towards the pole and arms are following it or trail behind it. This movement exhibits different shapes to the chromosome like I shape, L shape, rod shape, J shape, etc. based on the position of the centromere. So that is about the anaphase stage. Now let me talk about the fourth stage, that is telophase. In the telophase stage, the daughter chromosome moves towards the two opposite poles. As soon as these chromosomes move towards the opposite pole, the steps of the prophase is reversed. Like uncoiling or decondensation of the chromosome begins to form a long thin thread-like chromosome. The spindle fiber break down to form a tubulin proteins. The nuclear envelope is reformed. Nucleolus is reformed. So these are the main structure or the feature that takes place in the telophase stage. So that is all about the four stages of karyokinesis. So by the end of the karyokinesis, division of nucleus takes place or separation of the daughter chromosome takes place. Now let me talk about the second step of the young phase, that is cytokinesis. Cytokinesis means division of a cytoplasm or division and distribution of cytoplasm to the formed daughter cell. Cytokinesis in the case of animal cell is achieved by the formation of furrow or cleavage. This furrow or cleavage is formed in the plasma membrane. As you can see in this diagram, this is a furrow or cleavage which is formed in the plasma membrane. This furrow or cleavage deepens and ultimately it joins at the center, dividing the cytoplasm into two, or there is an equal distribution of the cytoplasm to the two daughter cells. So that is about the cytokinesis in the animal cell. Cytokinesis process in the case of plant cell is quite different. Because of the presence of cell wall, cytokinesis process takes place in a different way in the case of plant cell. In the case of plant cell, cytokinesis process is achieved by the formation of cell wall at the center of the cell. This cell wall formation takes place at the center of the cell by the formation of its simple precursor called as cell plate. So this cell plate is formed at the center of the cell, and this cell plate grows outward, which ultimately reaches the existing cell wall, thereby dividing the cytoplasm into two cells, or there is an equal distribution of cytoplasm to the two daughter cells. So that is about the cytokinesis process or division of a cytoplasm. So by karyokinesis and cytokinesis, a single cell divides into two daughter cells having equal number of chromosomes similar to that of the parental cell. So that is all about the mitosis process. I hope this video will be useful. If you like this video, please hit the like button and subscribe to my channel. Thank you.